Sakshi, you can start. Hello.
Hello, sir. Sakshi is not host. Avinash is the host.
Okay.
Yes, sir.
I mean to say you can start because speaker is available.
Yes. Hello, Avinash.
Hello.
You can start.
Am I audible? Hello.
Yes, you are.
Okay. So, a very good evening to all the participants. On the behalf of Just Agriculture, today I, Avinash Kumar Bhatia, doing PhD from Dr. Y.S. Parmar, University of Horticulture and Forestry, Nauni Solan, Himachal Pradesh. It is an opportunity to me, and I am happy as today I am the host for this great event for the students of agriculture, horticulture, forestry background of all over India. As this series will help and nourish the students of different disciplines, like in horticulture we have different disciplines about net, like vegetable science. In agriculture we have entomology, pathology. So every students from different disciplines will definitely get help and will success after viewing our this net series of 7 days. So I again welcome all the participants. Our respected first speaker, that Sir Bhaskar Reddy S. Sir is from Kerala Agriculture University, Thrissur. Sir, I welcome you in this next series. And basically, sir, from background genetics and plant breeding. That is a very what can I say that कि ये जो जेनेटिक्स एंड प्लांट ब्रीडिंग डिपार्टमेंट है, ये एक काफी बड़ा डिपार्टमेंट है एंड इट सपोर्ट्स मैनी नंबर ऑफ डिपार्टमेंट्स. So it is a backbone in agriculture also and in horticulture also. About net percentage, so sir has qualified net at very high score, that is 75%. So sir, I welcome you again in this net series, and now I hand over the session to you, sir.
Thank you. I'm happy that you have given me an opportunity to speak and share my experience regarding the preparation for ICAR NET. Okay, myself Bhaskar Reddy S. I have studied my UG from UHS Shimoga, Karnataka, and I did my PG from Chaudhary Charan Singh Haryana Agriculture University. And now I am pursuing final year PhD from Kerala Agriculture University, Thrissur. Okay. For now, importantly, coming to ICAR NET. Now, basically, I am from genetics and plant breeding. Let us discuss how to prepare, what are all the strategies to apply, and what are all the books we should prefer. Okay. Now, the date has given that from April 26, there will be going to conduct the exam. We don't know exact date, which subject having which date. Hardly today is 18, some 10 days is left. Okay. How to prepare within the stipulated time? You know, ICAR NET exam pattern, there are 150 questions and number of the time is stipulated time is only 120 minutes. Even you won't get 1 minute per question. Okay, hardly you will get some 45 seconds per question. Okay, you have to think it and you have to answer. Okay, this is what the pattern, that is 150 questions and 120 minutes. And each question carries 1 mark and negative is 1/3, that is 0.33. So while attempting the exam, we should be careful because it is not 1/4, because each and every exam like SRF, JRF, whatever CSR and all, there will be 1/4. But here negative marking is more, that is 1/3 of the marks it is going negative if you done mistake. Okay, coming to subject, the syllabus. In genetics and plant breeding, there are totally 8 units. One is general genetics and plant breeding, economic botany and plant breeding methods, genome organization and cytogenetics of plants, quantitative genetics and biometrics, and biotechnology aspects, and plant genetic resources, stress breeding, and statistical methods and field plot technique. These are the 8 units present in our genetics and plant breeding. The major books we should refer for this ASRB NET is for genetics and plant breeding, because we no need to study the advanced book, advanced topics. Basically, in ARS NET, they will ask the basic things. First of all, we should know the basic definitions of each and every aspects, because they will directly ask the definitions. Because when you are seeing the syllabus, whatever the words you are seeing, all the basic definitions you should know, each and every topic. Okay, but books you should refer. First I will tell the books and later we will talk about the strategies. Okay, books, first we have to start with the Punnan Singh genetics and Punnan Singh plant breeding. After that, is some short notes. Basic, we have to go from basic to advanced. Sir, am I audible?
Yes, sir, you are audible. Yes, sir. Yes.
First, we have to move from basic to advanced. First, we have to study Punnan Singh genetics and Punnan Singh plant breeding. Later, we have to move to the genetics by BD Singh and plant breeding by BD Singh. Okay, these are the major books we have to refer. Coming to cytogenetics and all, it will biotechnology and PGR and all, it will be covered in our BD Singh genetics and plant breeding book. No need to refer separately for PGR and cytogenetics, biotechnology and all, because few chapters already included in BD Singh. It is enough. Okay. Next, coming to quantitative genetics, you all know that for quantitative genetics, all will refer this quantitative genetics by Punnan Singh and Narayanan. It is a very good book. It is enough for the ARS NET exam. Coming to objective books, compulsory, because every time the questions are directly chosen from the objective books, that is objective genetics and objective plant breeding by B.K. Prasad and BDC. Okay, you all know there are 2 objective books, one for genetics and one for plant breeding. That is from by B.K. Prasad and BDC. Compulsorily, you should refer that book, objective books, because in appendices there are many tables and formulas given at the end. That is very you have to glance each and every tables. and important points given at the end of these two objective books. Okay, it is very easy to glance, because in books it is sometimes it is very difficult, but it is compiled and given in these objective books. Last time also there are many questions directly from that books only. And next, another objective book for quantitative genetics, Objective Quantitative Genetics by Punam Singh. You have to refer that book also. They have given some few definitions, some question and answer types, and multiple choice questions. Okay, directly there is a possibility of coming questions from that Objective Quantitative Genetics by Punam Singh. Next, for easy glance, you can refer Genetics Made Easy and Keynotes on Genetics and Plant Breeding. It covers all the portion, keynotes on genetic and plant breeding. It is easy, within 1 or 2 days you can cover the entire syllabus of this NET, because it is briefly given each and every aspects. This is very important. These books are very important for preparation for ARS NET. For coming to economic botany or crop breeding type, you can refer your UG notes. There is a UG notes that contains or your practical manuals which you are studied in UG. In that manuals, you can get the floral biology of each and every crop. That is very important. And coming to some varieties, because in every time in NET exam there will be questions regarding the varieties. Okay, that you can refer recently released varieties, some that may be uploaded in ICAR website. And another one PDF you can refer is that improvement, that is marker assisted plant breeding related released varieties. There is a one PDF released by ICAR. You can refer what are all the varieties or improved varieties that is released, developed through marker assisted selection. You can refer that also. Okay, these are the major books you can refer for the ICAR NET. Next, coming to strategy, how to prepare. Okay, first of all, before going to start for any exam, not only ICAR NET, even if you are preparing for SRF, like PhD entrance exam and all, first you have to look at the old question papers. There may be availability of old question paper. You won't get in any websites, but few are available in YouTube channels. Some are explained few of the question papers, and some are available in Telegram or WhatsApp group. Here and there it is circulating. Even I have some 5 years question paper also that I will share you. Okay, first of all, you have to refer not only not previous year question paper, because when I written in 2019 exam, the pattern of questions arriving is different, whereas in 2021 the pattern of questions arriving is different. They have asked whatever asked in that exam, it is totally different from whatever they have asked, but all are basic only. So when we refer 3 to 4 years question paper before starting the preparation, I am telling, okay, just glance the old question paper. What are all the questions they have arrived? Okay, after glancing, no need to worry about the answer for each and every question what you are referring the question paper. Okay, that later you can see. Just glance the question paper. Okay, then note down which are all the books you can refer. And another point is refer the syllabus. These 3 things first you should know. First, you have to refer question papers priorly, then syllabus, and then what are all the materials I have to study. Okay, because after seeing the question paper, you are already MSc student. Okay, you have, because MSc is the main criteria for ICAR NET, right? You are already in MSc if you are studying or you are finished anything. Okay, you have already know that which are all the concepts which will be there in which book and all. Okay, then you can easily prefer which books I can refer for NET. Okay, this is what the first strategy you should apply. First, refer the question papers, next syllabus, and just note down which are all the books I should refer. Then go on preparing from basic to advanced books. I have told you, first start with Punam Singh, then BD Singh. Okay, then this quantitative genetic books, and later objective books. Later, just for revision purpose, genetic made easy and keynotes on genetics and plant breeding. These are the books. Okay, this is how you have to start. Coming to general genetics and plant breeding, it will start with history. Okay, coming to history, there will be a classical genetics, molecular genetics, cytogenetics, all these parts they have given. In that, you have to, there will be scientist names, year, and in which concept they have worked, and what they have given in conclusion. Okay, these are all the points you have to remember. If you are coming to Morgan, okay, Morgan studied on which organism? Drosophila. In which year? What is the concept he has given? Okay, these like you have to study, because every time there will be some 5 to 10 questions regarding the scientist or year or in which concept given by these things, and few of the book names which was published by that scientist, that also they have asked. Book names also they have asked in the exam. So in history part you have to look after these things: the scientist, in which organism he have worked, which concept he have given, in which year. Okay, and whatever the books he was published. These are the major aspects in history you have to study. Next comes to cell structure. You know what are all the cell organelles and from where it is produced. Okay, Golgi bodies, endoplasmic reticulum, all these things developed from which cell organelle, and what are their functions in the cell. This is very, very important. Next, cell division. Cell division, mitosis, meiosis. Meiosis, you know the classifications and all, meiosis 1 and meiosis 2, and which stage, what are all the events occurring. Okay, majorly in meiosis questions will come. Okay, that is in prophase 1. There are 5 substages where crossing over will occur, where the pairing will occur. All these things you should note down. And another thing is, sometimes there may be a chance of asking the questions like number of chromosomes present in this particular stage, in coming to prophase 1 or metaphase 1, or in prophase 2 or metaphase 2. Okay, where reductional division occurs, where equational division occurs. In mitosis and meiosis, there is a coiling pattern, plectonymic and paranymic coiling. Okay, these things, and how the orientation of chromosome. In mitosis, chromosome orientation is different, and in meiosis, chromosome orientation is different. These things you should note down. Okay, next, coming to other chapters like linkage and Mendelian chapter, gene interactions, Mendelian laws. Study the Mendelian laws perfectly, and regarding Mendel also, because many questions will come: where he was born, what are his works, in which organism he has studied, what are his conclusions, why he is succeeded, why he is failure. Okay, all these things. And there are some formulas in coming, like number of phenotypes, number of genotypes. Okay, perfect F2 population, that is 4 to the power n. All these formulas, number of homozygotes, number of heterozygotes. Okay, these formulas you have to remember. Coming to linkage. Linkage definitions who give the concepts, and which are all the types, like coupling phase, repulsion phase. Here linkage problems is very, very important, because in coupling phase, repulsion phase, they will give 10% recombination. It is in coupling phase. Okay, then they will ask percentage of the gametes, like capital AB or small ab. Here one thing, important thing is, whatever the parentals in coupling phase will be recombinants in the repulsion phase. What are all the recombinants in coupling phase will be the parental types in the repulsion phase. This you should remember. When you understand this thing, you can easily solve the problems. Another thing is probability. Probability problems, surely they will come, that regarding the Mendelian aspects. For example, they have given 3 genes segregating each other, like capital A, small a. They have given one F1, that is capital A, small a, capital B, small b, capital C, small c. When if you self, what is the percentage of particular genotype, like capital A, small b, and capital C, small c? What is the probability of getting this genotype after selfing, that all the 3 genotype genes are heterozygous in condition? You have to apply Okay, you have to apply the probability rules. Okay, in probability there is multiplicative rule and additive rule, which rule it is already given in BDC nicely. You can refer there. Next, coming to linkage. After linkage, there will be mutation. First, you have in mutation, overall the scientists give the concepts, types of mutations, and types of mutagens. There is a physical and chemical mutagens, their mode of actions. Okay, these are all the things you have to study. And there is a classification of mutations. Okay, nonsense mutation, missense mutation, all these things that you should refer. There is a one complete page given that classification of mutations. You have to remember each and every line of that definitions of in that mutation chapter. Next comes the genetic or organization. Okay, DNA material, genetic material. Like who gave the double helix model. First, we have worked regarding DNA and all, and types of DNA present and their rotation and all. Okay, these things. And next, Chargaff's rules, different Chargaff rules and double helix model. Each and every aspects you have to remember. Next comes the replication. What are all the enzymes involved in the replications? What are their functions? What are all the proteins involved? What are all the subunits involved in replication, transcription, translation, and even RNA processing? All these things. Main thing is what are all the enzymes? What are all the proteins? What are the main functions? Okay, that is involved in replication, transcription, translation, RNA processing, and all. Okay, each and everything it is BDC is enough for that. Okay, this is about the genetics aspects. Coming to plant breeding. Plant breeding, you can basic concept from history, it will start. Okay, like plant introduction, male sterility, self-incompatibility, all these things. Main thing is you have to remember the examples. Like for each and every breeding, there will be different breeding methods. Like plant introduction is there, mass selection, pure line selection, pedigree, bulk, backcross. Each and every aspects you have to remember the examples. With some examples, they may ask the variety released from which breeding method, like that they may ask. And main you have to concentrate on breeding methods as what is the purpose of the breeding method, what is the useful of this breed, what the output the breeding methods it will give. Okay, 1 second. Sorry for the disturbance. Okay, coming to breeding methods, each and every breeding methods, what is the purpose, main purpose of that breeding method, and what is the output we are getting from that breeding method, and what are the advantages and disadvantages of that breeding method, and difference between like from mass selection to pure line selection. Okay, nowadays which breeding methods we are using, all these aspects we should remember. Okay, coming to like hybrid breeding, how they will be selecting the parents, because in quantitative genetics also there are different designs which is majorly used for the selection of the parents for further hybrid breeding. That you can study from one biometric in breeding also. They have clearly explained which design is used for what purpose. It is a selection of parents or knowing the diversity present in the populations, each and every aspects. Okay, in breeding methods. Here also major thing in breeding chapters, you will get biotechnological aspects at the end, and seed technology and plant genetic resources chapter. Okay, these 3 chapters you have to study thoroughly. Okay, because questions will come, because these 3 chapters is enough. Nearly 20 questions will be there regarding these chapters, because each and every aspect there is a syllabus regarding plant genetic resource and biotechnology and seed related questions also arriving in our ICAR NET. So these 3 chapters are also very, very important. And coming to advanced breeding techniques. Okay, advanced breeding technique, it will start from first molecular markers. Here from molecular markers you have to Now the classifications like dominant, codominant markers, okay, hybridization based and PCR based markers, okay, which is multi-allelic and biallelic marker, single locus, multi-locus markers. This is the classification you should remember. Next, coming to mapping populations. Mapping population, you know, mapping population majorly used for the mapping the genes. Okay, mapping population. There are different type of mapping populations like F2 mapping population. Okay, F2 derived F3, RILs, BILs, NILs. Okay, magic population, NAM populations. There are different mapping populations, and main thing here you have to remember how they are produced. Mapping population, okay, they are mortal or immortal, which is the best mapping population, which is for the mapping in QTL mapping, which is the best mapping population, that is RILs, recombinant inbreed lines, which are developed through single seed descent method. Okay, like this, it is user friendly and it is best method. How like this, how they are developed, what are how many parents, like biparental mapping populations are also there, multi-parent mapping population like NAM and magic populations, they are the multi-parental mapping populations. And what are the purpose and what are the advantages and disadvantages of these mapping populations? After that, QTL mapping, how they are doing genotyping, how they are, what are all the steps of QTL mapping. Next, it will come, next will be the association mapping, RNA interference technology, antisense technology. Now the genome editing tools like ZFN's talents, CRISPR, base editing, different tools which are used for this advanced breeding. And marker assisted selection, marker assisted backcross breeding, and marker assisted recurrent selection. These are the things, but no need of studying or depth. Basic things you have to cover in these concept, advanced topics. Okay. In coming to genetic engineering and biotechnological tools, like tissue culture also very important, like somoclonal hybridization, micropropagation, which are all the plant material you are using for somoclonal hybrid, somatic hybridization or micropropagation, like that. And what is the use of tissue culture? Okay, for like sometimes they may be going for chromosomal studies, sometimes they may go for double haploid production. Okay, like this you can study. Next, coming to genetic engineering aspect. Genetic engineering, how, what is you mean by transgenics, how they are produced, what are all the different methods to transfer the genes, what are all the enzymes you are using, vectors you are using, vector classification you have to know, different type of vectors and the size inserted in that particular vectors, that you should know. Okay, vectors and the enzymes, restriction enzymes, their sequence is very important. There may like 4 base cutter, 5 base cutter, 6 base cutter of restriction enzymes. Classification is restriction enzyme 1, 2, 3 are there. Commonly used restriction enzymes 2, and their restriction site, there is a sequence of restriction sites for particular enzyme. They will ask, there is a 1 kb circular or linear DNA. Okay, they will ask, there is a 1 kb of DNA, you are having a 4 base cutter, but enzyme, how many fragments will be formed? Okay, how many fragments are formed from 1 base pair, 1 kb DNA, circular DNA, number of fragments will be produced by 4 base cutter. Okay, how many? 4 base cutter means there are how many nucleotides? 4 ATGC, right? For 4 base cutter, 4 to the power 4, that is 256 base pair. At every 256 base pair, there will be a cut in that 1 kb circular DNA. For 1 kb for 1 cut, then only 1 fragment will be formed. You have to 1 kb means like 1000 base pairs divided by 256, you will get the number of cuts. Number of cuts equals to number of fragments in case of circular DNA. Whereas in linear DNA, it is +1. In linear DNA, if you cut once, you will get 2 fragments. Whereas in circular DNA, if you cut once, you will get only 1 stretch of DNA. So number of cuts equals to number of fragments in circular DNA, whereas in linear DNA, it is +1. Okay. If they have given 5 base cutter, what you will get? There will be how many base pairs? Like 4 to the power 5, that is 1024. Whereas if they have given 6 base cutter at 4096 base pair, it will cut. Every 4096 base pair, the enzyme will cut. So based on this, what they have given, size of the DNA and which restriction enzyme, it is 4, 5 or 6 base pair cutter, and they will list number of fragments or number of cuts. It depends. So you have to remember, study the question properly. Okay. Next, coming to plant breeding for stress resistance and nutrition quality. That in BDC, there is a abiotic stress and biotic stress and water chapters, and quality breeding also. There you can study clearly what are all the mechanisms for drought, salinity, cold tolerance, all these things. And for insect resistance and pathogen resistance, what are all the mechanisms and what are all the breeding methods is used. Okay. In nutrition quality, the protein quality and oil quality, and what are all the what is oil content and protein content in different crops. These are very, very important. Plant genetic resource and their regulatory system, variety release and seed production. It is also chapter present, plant genetic resource and seed variety release and seed production. These chapters are available. It is very simple and you can read easily. Sometimes there may be asking like hybrid seed production, isolation distance and purity percentage, all these things, seed related aspects they may ask. And in coming to PPVFR Act and all, you know, the farmers' rights, breeders' rights, exemptions, all these things you should refer. Coming to statistical methods and field plot techniques, the basic thing here you have to refer is your UG notes. UG statistics notes is enough for this last unit, that is statistical methods. Or some already some like 38-page notes is circulating in Telegram and WhatsApp also. It is crisply, I think from IASR someone have done and they have sent in WhatsApp groups and Telegram groups. If you got that one, you can refer. It is enough for glancing. Within 1 hour you can glance the notes. It is very, very useful and informative. Okay, these are all the things. In for example, I forgot, like in breeding methods, how at what stage the selection, like come in bulk method, we will selection when the population attain F6. Okay, whereas in pedigree we will start selection from F2, like that. What is the main purpose of backcross breeding? Main purpose of backcross breeding is only a gene transfer? No, right? Backcross is majorly done for the recovery of the recurrent parent genome. Okay. Sometimes there may be option, transfer of gene, recovery of genome, both, but both. Okay, both recovery of genome and transfer of gene. Okay, this is what you should understand. Like these questions may come. Whereas in coming to population improvement methods, you can, you know, GCA and SCA. What do you mean by GCA? General combining ability. What do you mean by specific combining ability? Next, what do you mean by simple recurrent selection? Recurrent selection for GCA, SCA, reciprocal. What is the difference between them? What is the use? What is the difference and what are the uses of these different recurrent selection schemes? And what do you mean by composites? What do you mean by synthetics? How they are developed? Okay. There is one question last time they have asked minimum number of population. Why there will be minimum number of population? Like we can't use less or we can't use more number of inbreeds for the development of the synthetics, because minimum we can take it as a 4, maximum 10. Okay, if you go on increasing the number of inbreeds for the development of synthetics, what happens? The general combining ability between the first inbreed and the last inbreed will be less. That's why there is a decrease in the yield. So there will be minimum. This is what the reason. Why we have to use some minimum 4 and maximum 10, not more than that, not less than that. Okay, so remember, because the general combining ability between the first inbreed and we are taking 11th or 12th or 15th inbreed, that will be less. Okay, that's what, because developing the synthetics and all, we will go for checking the general combining ability like this. And as the number of inbreeds increasing, the yield level will increase. Though decrease in the yield level is slow in synthetics, but there is a limit for everything. Okay, like that. These are the things. And problems, recovery of in for example, percentage of heterozygous individuals, percentage of homozygous individuals. Okay, these are all the formulas you can see in breeding. What are all the formulas? All these formulas and all things is in this objective BDSing and BK Prasad. You can refer that objective books at the end. They have given all the formulas. You can refer that. Okay. And next important thing, problems will come from this population genetics, Odi-Winberg law. That is basic thing, P plus Q equals to 1, P square plus 2PQ plus Q square equals to 1. If you know this basic thing, you can easily solve. Okay, these are the things what you have to prefer, what you have to study. Okay, next coming to how you should attempt the exam, because only 2 hours left. First of all, when you are going to attempt the exam, there are 150 questions. What happens sometimes, there will be starting when you are attempting the question, you don't know, because I have also faced a lot of time. First 5 to 6 questions, even you don't know nothing, you don't know. Suddenly you will got frustrated. Oh, I have studied well, but I don't know I don't know nothing to worry, because 150 question, we need to clear the exam only 75%, right? Sorry, 50%, only 75 marks we need. So nothing to worry. First, just go on attending whatever you know, 100% because don't take chance at the first attempt. Okay, first you go on attempting, look at the whole 150 questions which are very easy, you attempt with surety, 100% surety. Okay, after that you can go for next chance. This, like for example, out of 150 you may attend some 50 to 60 questions directly by seeing the question paper. Okay, within some 20 minutes you will attempt easily. And next you have to go for the question elimination round. Okay, because some questions but you are in confusion mode, because maybe this or maybe like 50-50. That time you have to apply elimination. Okay, you have to eliminate the options, then only will approach towards the answer. Okay, that is very, very important. Don't frustrate, nothing to worry, you will easily clear whatever all the books I have referred. If you glance it once, you will easily clear. Questions will come from that only, not from any other. Okay, just main thing is no need to be in a cool mind, then only you can. Any exam, if you are in a cool mind, then only you can attempt the exam very nicely. If you are taking tension, even because when you take tension, what happens, whatever you reading the question, you will read wrong. If they have given one question, it is not, they have given not, but you will read, you skip that not and you will read directly question and you will answer on another thing. Okay, there will be very it is incorrect, they will given, find out the incorrect statement or find out the incorrect among these options, like that. But you don't read incorrect, only correct. Because what happens now, some questions will be excitement, easy questions you will see, but you don't read question properly. That is the big mistake many people will do. So read the question carefully 1 or 2 times. Okay, don't be so much excited or don't be so much tense. Be cool, read question properly 1 or 2 times, and also the options. Sometimes option will be similar. Okay, that time similar means not same but somewhat related. Okay, that's why don't be confused. When you read 1 or 2 times, then you will get clear question and you will answer perfectly. Okay, first attempt, easy one, whichever first attempt that one. Later you can go for eliminating the answers. Okay, if you eliminating the wrong options, you will reach the correct one. That is the best thing you can do, because if you don't know the answer also, by somewhat residual knowledge, you can eliminate the wrong keys, and you can suddenly, okay, you will be already even there will be mind that this is the right answer, but you don't have surety. So when you eliminate the options, then you can easily attempt the answers. Okay, out of 150, better you can cross, like attempt some 100 plus if it is tough. If it is easy, you can go for more than that, because in first attempt it was tough. I attempted some 90 plus, but there is so many wrong. That's why I didn't clear that time. Also, I didn't study nothing, just I studied for SRF 6 months before. I didn't touch the book, just attempted like that. So that time I am not able, just by 2 marks I have missed it. But next time, yeah, I was perfect that every time. Even I have cleared CSR, JRF net. That time also, in third attempt, I have cleared CSR. There also same thing, what I have mistaken in first 2 attempts without studying properly and all, that I have rectified in my last attempt, and I have cleared that one. So same thing, nothing to worry. Whatever the mistakes you are doing, because many exams you are not only ICAR, it is your first attempt, nothing worry, but you have attempted some other entrance exams, ICAR, JRF, SRF, like that. Whatever you have done the mistakes, don't repeat each and everything every time. Okay, just rectify your mistakes and attempt the exam. Okay, better some nearly some 100. It is better if the paper was tough. If paper was easy, it is left to you how much you can attempt. Okay, this is all about the how you can prepare for the ICAR NET Genetics and Plant Breeding. You can contact me. I may give you my contact number also, because from 5 years I am guiding JRF and SRF students. This time I am not able to, because of my last PhD thesis submission. That's why I am not able to take SRF and JRF coaching. So you can contact me also. My contact number is 8277465941. Okay, you can message me and you can contact. If you need question papers and some materials also, I can share if you contact me also. Thank you for Just Agriculture for giving me an opportunity to speak. Thank you.
Thank you so much, sir, for your kind time and your kind word, your kind lecture. Again, from Team Just Agriculture, I would like to thank you, Bhaskar sir, for this and enlightening lecture on right way to how to crack the ICAR-ASRB-NET examination. I hope the student from the genetics background will definitely get an edge in their exam from this lecture. Thank you so much, sir. Thank you, sir. Thank you.
Thank you.
So for the second session, we are very glad to have Dr. Swati Bhimraji Gaikwad. Ma'am is basically from Crop Physiology Department, and right now, ma'am is Assistant Professor at Ram Krishna Bajaj College of Agriculture, Pipri Vardia. Welcome, ma'am.
Thank you.
Yeah, ma'am, you can start your this lecture series.
ठीक आहे बरोबर हा ऍप बनवली म्हणजे त्यांनी
Participants are 4 only. Shall I wait for some more time?
No, ma'am. You can start the session, ma'am. This will also go through live on the YouTube. Participants may join there.
Oh.
Yes, ma'am.
So I have some slides presentation with frequently asked question in ARS. ARS means and NET examination. So I'm working here in Bajaj College of Agriculture. I'm Dr. Swati Gaikwad, and did PhD from US Harvard in 2016. And I have 3 to 4 question papers. These are memory based. I have written after examination, after completion of examination, I have written in home and answer to that question. So many times this question is asked in the examination. So you can see the ARS combined. This was the combined notification for ARS and NET. So the question paper for prelim and ARS means was same. ARS prelims was same. So the first question, it was a top sickness of a tobacco. It is a boron deficiency. So top sickness of tobacco, it is the physiological disorder of tobacco plant, and it is due to the boron deficiency. This is very famous disease, that is Khera disease of rice. So the question is asked from this physiological disorder of the different plant, like different crops which are suffered due to some disorder like mineral deficiency, or you can say that the climatic changes or disturbances are giving a more dose, more fertilizer dose, or improper fertilization, or due to faulty interculture operation. So like that, there are many processes which are many factors responsible for the physiological disorder. So the question from physiological disorder, compulsory they used to ask in examination. So 1 or 2 questions it might be there in different examination, but particularly in NET prelims and ARS examination, they used to ask the questions from this section. So what are the different physiological disorder? Which deficiency of mineral element is responsible for different physiological disorder, or any environmental parameters or toxicity of any mineral nutrient. So these are the different factors which are related to the physiological disorder. But these 2 are very famous question. Toxicness of tobacco, it is due to the boron deficiency, and Khera disease of rice, that is a zidane deficiency. Hormone not synthesized by amino acid. So salicylic acid, it is we call it as a hormone. In taste and figure, you can see that the salicylic acid, jasmonic acid, fumaric acid, and the trichontanol, these are acting as a hormone. So in hormone biology chapter, you can see that the salicylic acid, it is also regulating the different processes in the plant. So the hormone not synthesized by amino acid. So for the hormone, there are different biosynthetic pathway, majorly amino acid relating to the biosynthesis of hormone, like your ethylene. It is a precursor element. It is methanoid, and methanoid, it is one of the amino acid. But here, so among all the hormones, salicylic acid, it is one of the hormone which is not synthesized by amino acid. So it is tricky somewhat, but the answer, it is salicylic acid. Which vitamin related to the human eye disease, that is a beta carotene? It is a precursor element or precursor pigment for the vitamin A. And vitamin A, in case of a vitamin A deficiency, there is a human eye diseases may occur. So the vitamin related to the human eye disease, it is a beta carotene. Prokaryotic differs from the eukaryotic due to there are different options: chloroplast and mitochondria, chloroplast and cell wall. So ultimately the answer, it is none of this, none of the above. Lodging in cereal controlled by silicon, and the lodging in rice. So additionally, I am giving some points here, that calcium, the control of lodging in rice, it is due to the calcium. But in cereal crop, other cereal crop, it is might be controlled due to the silicon. So like this question, these are the some question. So there are series of questions I have given here, and these are very important questions for the NET and ARS prelim preparation. So many times they are repeated only. And some new question also I have added here. So in the next slide, there are some questions like which gas related to the fruit ripening? And the answer it is ethylene. So all the gases except methane. This question, see this question. So ethane, ethylene, and acetylene. Acetylene, it is structurally similar to the ethylene. And ethane, it is also nothing but the ethylene only. But the methane only, except methane, all the gases are related to the ripening only. But you have to select the ethylene. That is, it is a fruit ripening hormone, ethylene. So ethane and acetylene also promoting the fruit ripening, but the answer it is a ethylene. Agent Orange chemical. So in the World War, this herbicide, this Agent Orange has been utilized, and that is a 2,4-T, 2,4-T, like that. That is a herbicide. The Agent Orange, it is nothing but the auxin group of hormone, and it is used as a herbicide. So this is what the herbicide from the hormones at the higher concentration we can use as a herbicide. So laughing gas NO2, this is very common question. Sea level rise due to the global warming, 3 mm per year. This is update data, means from some research article I have taken this answer. So in the option, it was a different question, different answer. That is not a 3.2, 3.3 like that. This is but I have written here 3 mm per year. This is from recent articles. Temperature increases due to the global warming. That is per year 0.8 degrees Celsius. It is a global warming temperature. UP depletes the ozone from which layer of atmosphere? So answer it is nothing but the stratosphere. So stratosphere, it is the region where UV deplete the ozone. So the ozone, it is a protecting layer. Paclobutrazol response, it is a plant growth retardant. This is a plant growth retardant, paclobutrazol. To promote flowering in mango, spraying of gibberellic acid at 50 ppm. So there are different options, that is carbaryl, ethefone, GA, and none of the above. From this, the gibberellic acid at 50 ppm cause delayed panicle emergence, increased number of perfect flower, and increased pollen viability in the mango. So answer it is that the GA at the rate 50 ppm. It is promoting flower in mango after spraying of the GA. So there are 2 types of germination, actually 3 types: epigeal, hypogeal, and viviparous germination. But the epigeal germination, the first stage, sometimes they used to ask that what is the first phase of a germination. Sometimes they ask that what is the seed, how the seed undergone the germination, what is the first phase when it undergone the germination. Here they have asked the epigeal germination, the first stage. What is the first stage? That is a imbibition. So imbibition, it is a first phase where the different hydrophilic colloids, which are responsible for absorbing more water, oxygen, and it swollen the seed. After that, the radicle and pleuble will get at the time of a germination. So imbibition, it is a first phase of a germination stage. Then qualitative short day plant, it is a Xanthium strumarium. What is the background of this question? I have to explain here. Quantitative short day plant, qualitative short day plant, like short day plant and long day plant and day neutral plant. These are the classes of a plant. according to the photoperiodism occur in the plant. So it is the phenomena related to the flowering. But the qualitative short day plant, it's meaning that the short day it is required to flower the plant. So particularly it is a Xanthium strumaria. So you have to learn about qualitative short day plant, what are the quantitative short day plant, and sometimes they used to ask like qualitative short day plant and the facultative short day plant. They are nothing but the similar. Qualitative and facultative, they are nothing but the same. The answer it is only Xanthium strumaria. There are many examples which are comes under the qualitative short day plant and the facultative short day plant. So likewise in the long day plant and the in the long day plant, we can see that the facultative long day plant also there. So how we can simplify this? We can say that the qualitative long day plant are nothing but the facultative long day plant. So likewise, the compulsory they used to ask the question from this section, the photoperiodism, and they used to ask the example. They used to ask the scientist contribution. They used to ask the different variety. They can ask the biloxy variety of soybean, which is the short day variety of a soybean, like biloxy variety of soybean, and then Maryland Manor is the tobacco variety, which are comes under the photoperiodism section. So photoperiodism chapter is very important. You have to go through this, and 2 or 3 questions it comes from the photoperiodism section. So this is one of the questions from that section only. The chromosome contain protein and DNA organized into gene. Option was like that. Chromosome contain DNA, RNA, and DNA, protein and DNA are organized into gene. So this is the correct answer. Protein. Also protein and the DNA organized into the gene. So this is a proper answer for the for this question. This is a calculatory part, like you know that our water potential is equal to solute potential minus pressure potential. So you can calculate this with this formula and you can answer this. But you have to take care about that water potential. It is always negative, and the most translocated mineral element in the plant. So you can classify the mineral element in the plant like mobile element and immobile element, and translocating element and non-translocating element, like your calcium. Calcium, it is a immobile. So in mobile, you can see that the nitrogen, like there are different elements which are mobile, and these are the most translocating element in the plant body. So you can classify it from the mineral nutrition section. They used to ask about the Ernan and Stout criteria of essentiality. They can ask the functions of a different mineral element. They can ask what are the elements, which elements are the cofactor of a particular enzymes, like your potassium, then calcium, then Zn. So one example I can say that the Zn, it is a cofactor of a carbonic anhydrase, and carbonic anhydrase, it is a very important enzyme. And you can see that in the next coming slide, carbonic anhydrase, question on carbonic anhydrase they used to ask in the examination. So you have to take care about that from the mineral nutrition chapter. The functions of a different elements, Ernan and Stout criteria, question related to the hydroponics, question related to the aquaponics, then question related to the different deficiency symptoms and the uptake of a mineral nutrient. Then from the VAM, that is vesicular asbescular mycorrhiza, and these questions are related to the different physiological processes. Which mineral element are different physiological processes? Like the famous question, it is from the photolysis of water, which is takes place in the PS2 mechanism of the photosynthesis, where the plant required chlorine and the manganese responsible for the catalysis of water. After that, the electron get transport into the PS2. So this is what the question comes from, regularly comes from the mineral nutrition chapter. Scientists from the senescence physiology. I didn't get any scientists from senescence physiology. And this are related to the aging process, programmed cell death. The question, many question comes from the senescence physiology also. In senescence chapter, what is the aging, what is the generatology, and the aging process, that is a programmed cell death. Likewise, they used to ask, that is monocarpic senescence. So one question from monocarpic senescence, that in which crop it is occur, that is annual, and this answer, it is directly this question and answer both are written in the Tez and Ziegler book, that is page number in 485 पेज नंबर पे ये क्वेश्चन का आंसर दिया गया है, मोनोकार्पिक सेनेसेंस इन विच क्रॉप इट विल अकर, दैट इज ए एनुअल क्रॉप. Then the flower of a papaya, option it is a staminate, pistillate, hermaphrodite, and none of this. So answer it is a hermaphrodite flower. Everybody knows that the fruit of mustard, it is called as a silica. That is a silica type of a fruit. Mimicking effect of a nitrogen. Mimicking effect of a nitrogen. It is a molybdenum. Both the nutrients cause similar deficiency symptoms. In case of a nitrogen deficiency and in case of a molybdenum deficiency, same symptoms can occur due to the nitrogen and molybdenum deficiency. So answer it is a molybdenum. So similar effect, this is a mimicking effect. So rhizobium fix nitrogen by using the enzyme nitrogenase. So everybody knows this nitrogenase enzyme, and it is regulate the biological nitrogen fixation, which is carried out by different bacteria. And from that, the rhizobium, it is one of the organism, one of the bacteria which fix the nitrogen by using the nitrogenase enzyme. Yes. Which element is a part of a nitrogenase? So nitrogenase, it is the enzyme. In previous slide, I have explained you what is the function of nitrogenase. So the minerals like iron and the molybdenum, these are the component of a nitrogenase enzyme. And particularly nitrogenase, it is the enzyme which can locate into the chloroplast and the plastid of the leaves and the root. So likewise, the biological nitrogen fixation, where the nitrogenase it is having very important role, playing important role into the nitrogen fixation. Then light reaction, which element? It is a acceptor element. So first, when the light trap on the plant, light fallen onto the plant, so the first protein, that is a thiophytin protein, which accept the light and regulate the different processes related to the photosynthesis. How many ATP required to oxidize one CO2 molecule by Kelvin cycle? It is 80. Cycle dominance is a famous question. It is contracted by auxin. Then dormancy of seed and embryo related to the hormone, that is a ABA. So from the hormone biology, hormone physiology, they used to ask about what are the precursor of a different hormone. Like the earlier I have given example of a methanoin. Methanoin, it is an amino acid, and amino acid, that methanoin, it is a precursor element of a ethylene. So likewise, tryptophan. Tryptophan, it is nothing but the amino acid, and it is responsible for synthesis of a indole acetic acid. Indole acetic acid, it is a natural oxide, and responsible for rooting of a different cutting of the plant. So it is related with the related with the apical dominance, that auxinate is related with the apical dominance. So likewise, they used to ask about the what are different precursor of the different hormone, what are the biosynthetic pathway, how it will be regulated, what are the different function of a hormone, then signaling of the hormone, and the role of a hormone in different physiological processes. So from this section, compulsory they used to ask the question from the hormone physiology. Then the development of an embryo without fertilization is parthenocarpy. Function of a Golgi body, it is a storage of polysaccharide. Sometimes we call it as a transport of sugar also. So the function of a Golgi body, it is a storage of polysaccharide. It is given in a Vicky Jain book. So you can see that the book where the function of a Golgi body is clearly mentioned that it is related with the storage of polysaccharide. Packaging, food packaging organelle, that is a Golgi body. This is also mentioned in the Vicky Jain book. Suicidal bag of an enzyme, that is a lysosomes. So everybody knows that why we call it as a suicidal bag of the enzyme to the lysosome, because from our body the RBCs not having the lysosomes, and the life cycle of RBCs, you know that the 120 days, and the cell which having the lysosomes which can recover fast. So whatever the dead tissue they can get died due to the lysosomes. So lysosomes, it is very, very important for the body. So because they have enzymes, and such enzymes which are important for removing the dead tissue from the body. So that is why we call it as a suicidal bag of the body, that is a lysosomes. So the different functions of function of a different organelle, what are the marker enzymes of an organelle, and what are the cell organelle, what is the structural and functional features of the different organelle. So there are many questions comes from the cell organelle chapter also. So the question like what is which is the marker enzyme of a peroxisome? So I think so everybody knows these things, that peroxisomes, the marker enzyme for peroxisome, it is a catalase. So what is the role of catalase then? Catalase, it is a breaking the H2O2, hydrogen peroxide, and convert into the water, and it reduces the reactive oxygen species. H2O2, it is what? A reactive oxygen species which totally broken down with the help of catalase. So catalase, it is present in the peroxisome. That is why the catalase is a marker enzyme of a peroxisome. So likewise, they used to ask about what are the functions, how the structural development of a different organelle, and the functions of a different organelle. So like this, they used to ask in the examination from the cell organelle chapter.
Hello.
Yes. Yes. Sorry for disturbances. Okay. So what is Corenberg enzyme? That is DNA polymerase, and this is the enzyme which are related with the TAP, that is PCR, and polymerization of a DNA. So the question from biotechnological portion, they used to ask like PCR, then the gene transfer method. specifically agrobacterium tuberculosis AGBT technique of the gene transfer, and particle bombardment method, gun method, and gene expression, central dogma, like transcription, translation, protein synthesis, and DNA polymerization, DNA replication, and the marker, DNA marker, like RFLP, RAPD, then AFLP, then SNP, and all the markers. I think so. The question like what are the co-dominant marker and dominant marker? It is very common question of ARS. So they used to ask many questions from the biotechnological portion, and some portion from plant tissue culture also, like the media composition, the hormone proportion, like auxin and cytokinin proportion for the callus induction, and different stages, like which hormone relates the rooting, which hormone relates the shooting, and the scientists related with the media and scientists related with the tissue culture. They used to ask in the examination. So there are many questions they used to ask from the biotechnological portion, and you had to learn all the details about the about whatever I have explained now, like the marker, PCR, then gene silencing, then different marker like RFLP, AFLP, RAPD, SNP, and SCAR marker, then RT-PCR. Likewise, they used to ask. Then triglyceride synthesize in which organ? That is the endoplasmic reticulum. Stress-related protein. It is a heat shock protein. Heat shock protein present in the plant are expressed at the time of a stress. So different kind of a stress, that is the heat stress, temperature stress, where the heat shock protein will be synthesized, and there are a different range, like HSP 100, HSP 70, HSP 60, like that. So heat shock protein, it is a stress related protein, and sometimes they used to ask stress related amino acid. So I guess that the answer, it is a proline. So stress related protein, it is a heat shock protein, and stress related amino acid, it is a proline. So proline, it is one of the heterocyclic amino acid, and it is related with the stress responsive proline. So at high concentration of a heat shock protein and the proline, plant can relieve from the stress. So what are the stress? Like oxidative stress, drought stress, then biotic stress, abiotic stress, then temperature stress, freezing stress, chilling stress, and the different stress. From there are different questions they used to ask from the stress physiology also. So you can read the Dwivedi and Dwivedi book for the stress physiology, where you can get the answer of many questions related with the ARS, and the book is very nicely written. So you can refer that book, Dwivedi and Dwivedi. The author, it is Dwivedi, that stress physiology. Then under stress, which saturated or unsaturated fatty it is increasing. So under stress, ultimately the answer, it is a unsaturated fatty acid. When the plant it is under stress, unsaturated fatty acid, it is a increases in the plant. So the banana respire more due to its climacteric nature. So don't forget to read the climacteric group of fruit and non-climacteric group of fruit. So what are the examples of a climacteric fruit and what are the examples of non-climacteric fruit, that we have to read. And the question, it is compulsory asked from this section. What are the examples of climacteric fruit and non-climacteric fruit? So you can refer the Wikigen also. There they have given the different climacteric fruit example and non-climacteric fruit example. These are classified due to the acceleration of a respiration at the time of a ripening process. So in climacteric nature of a fruit, there is a acceleration of a respiration, and the banana where this process it is regulated. So banana respire more due to the ripening in fire because it is bearing a climacteric nature. Then tendril formation, due to which hormone it is occur. So tendril formation, you know that tendril, it is a modification of a leaf, modification of a stem and the leaf, and where the tendril will occur. You can see this type of a organelle in this type of organ into the pea plant and the passion flower also. So this is due to the GA, gibberellic acid. One question from the velamen, that is velamen. It is a spongy tissue in the root system, meant for the respiration. Not a respiration. Velamen, it is a spongy tissue into the root system. So SOS, what do you mean by SOS? SOS, it is a salt overly sensitive, related to which stress. So ultimately, salt overly sensitive, but long form it is not given in that question. Only SOS they have given. So what is that? Salt overly sensitive. So it is a salt stress. So salt stress where the SOS factor it will be expressed in case of a salt stress. Gas inhibit ethylene. Which gas inhibit the ethylene effect? That is a CO2. You can see this, the biosynthetic pathway of ethylene, where you can get the idea about how the CO2 will regulate the effect of inhibit the effect of ethylene. Can plant produce which acid into the in night? So very common question. Oxalic acetic acid and malic acid. So both are the organic acid produces into the crocylin acid metabolism plant. Then active form of a phytochrome, that is a PFR. There are two forms of a phytochrome, PR and PFR. This is PFR for ranges far red, and PR it is for the red light wavelength. So, but the active form, both are interconvertible to each other. PFR will convert it into the PR, and PR will convert into the PR at the different nanometers of a wavelength. But you have to learn about the biological active form of a phytochrome, that is a PFR. Then plastochrome development means. So answer, it is a time interval between leaf initiation, or sometimes they used to ask the time interval between the leaf development, or sometimes they used to ask 3 times this question has repeated, and answer it is differently interpret there. The time interval between leaf initiation and development of a leaf primordia and leaf development. These are the 3 questions, 3 answers for the plastochrome development. So the answer it is modified, but the meaning it is same, like time interval between leaf initiation and leaf primordia development and leaf development. The answer it is nothing but the same, that is a plastochrome development. Membrane integrity index, it relates what? ये क्या indicate करता है? So membrane integrity index, it relates the function of the membrane. It is a proper function of a membrane. So due to some stress, there is a leakage from the membrane and destruction of a membrane integrity due to stress. So it relates the stress physiology. The question from stress physiology on the membrane integrity index, which can mention the functions and regulation of membrane properly, and this is why this is from the membrane integrity index. Mutant lack function due to the foreign structure. So I have written the gene silencing, but I think so we have to search for this question. So mutant lack the function due to the foreign structure. So please read the gene transfer method or the mutagenesis or the mutant. Mutant which lack the function due to the foreign structure called what? So we have to see this question in depth. Callus differentiated in different form called as a redifferentiation. So there are dedifferentiation, redifferentiation, and these are the processes which occur into the plant tissue culture. And in case of organogenesis, so morphogenesis and organogenesis. So this question, it is comes from the tissue culture methods, tissue culture techniques. Within 666 to 670 nanometers flowering response due to which pigment, that is it, phytochrome. So phytochrome, we can say that it is also the pigment, it is also the protein, and it is related to the flowering. Global warming potential of a methane, that is a 23. So this is from the climate change portion, and the global potential of different gases we need to understand. That is methane, then CFC, then nitrous oxide, sulfuric, what all the gases which are related with the global warming. So they are comes under the responsible gases for the warming, and from that section, the different gas which have a different global warming potential. From that, the methane, it is one of the gas, and this methane, it is having a global warming potential, that is a 23. So it is different for the different gas. So go through this this section from the global climatic changes, and you have to see the different global warming potential for the different gas. So any question comes from this section for the different gas. Aflatoxin produced from Aspergillus flavus. So aflatoxin, it is related to the groundnut crop, and it is due to the Aspergillus flavus. Vines cutted to maintain the shoot from main axis, middle, base, and the branches. So vines cutted like grape crop. So it can be cutted from the branch, and the pruning practices, it can be a part of a grape cultivation, vines cultivation. So then Taq DNA polymerase, you can use into the PCR reaction. One more question related to the Taq DNA polymerase, that is from which bacteria it is isolated. It is a Thermus aquaticus, and it is a thermophilic bacteria. And this Taq DNA polymerase, which is isolated from this particular bacteria, and it is used in a PCR. Self-pollinated crop, cross-pollinated crop. From this section, okra, they have asked the self-pollinated qualitative short day plant. Again, this is a repeated question. Plastochrome, sensitive to light, that is its infrared and far red. Phytochrome, phytochrome, it is a pigment protein which is sensitive to the infrared and far red light. LAI limit, it is 2. This is from the growth analysis. Then CGR and LAI relationship, it is equal to NAR. Then total output of photosynthesis and respiration, we call it as a net production. Carotenoids converted into the anthocyanin. This is very tricky question. I have read all this material, that is biosynthetic pathway of carotenoids and anthocyanin, but we have to learn more for this. This question, it is very tricky for me. Most of the reactive oxygen species in which organelle? So it is in chloroplast, mitochondria, and peroxisome. So peroxisome, I have explained earlier how the catalase, which is helpful for the degrading reactive oxygen species. Then two protoplasts are fused with the chemical, that is a PEG, polyethylene glycol. Calcium sulfate commercially in the form of a dolomite. Then stomatal opening, it is due to the blue light. Then chemical dormancy induced due to the phenolic compound. So it can also induce to the ABA hormone. It can also induce to the ethylene also. But here, both the options are not there. It is the phenolic. Phenolic are the chemical which can induce the dormancy into the seed. So from the seed technology, seed physiology, you have to learn about the different types, different classes of a seed. What is the structure of a seed? What is the storage practices of a seed? What is the moisture percentage of a different seed, which are feasible to store the different seed, and the post-harvest handling of the seed. Then the physiological phenomena takes place into the seed germination, factors affecting seed germination, and different successive stages of the seed germination. So all these portions comes under the seed physiology, and you have to go in detail for the preparation of a net. And the question they are asked from the seed physiology section. This is the paper, that question paper, and 2013. Transpiration pool theory, it is given by Dixon and Jolly. Then this Dixon and Jolly, also the question, it is asked like that, cohesion and adhesion theory, who given this theory? The answer, it is saying that Dixon and Jolly in 1984. Inductive enzyme, that beta galactosidase enzyme, it is when the substrate it is there, for example, the galactose, and alpha galactosides. So these are the different substrates where the inductive enzyme, it is a beta galactosidase. Cell organelle associated with the triple P. So plastid and cytosol, pentose phosphate pathway, where you can see that the ribose, and xylulose, these are the high carbon containing sugar, that is a pentose sugar, which is the process related with the plastid and the cytosol, where it is up. Then glycolate associate with the conversion of acetyl CoA to succinate for synthesis of a carbohydrate. So you know that glycolate cycle, it is a modified version of a TCA cycle. So particularly into the glycosome. So they are produced not a alpha ketoglutarate, they produce the glycolate, and this glycolate, it is related with the oilseed crop, where you can see that all the process similar to Krebs cycle from the acetyl CoA to succinate, but in between that the glycolate production will be there. So that is why the glycolate cycle, it is associated with the acetyl CoA into the succinate formation for the synthesis of a carbohydrate. Marker enzyme for the ripening process, that is a polygalacturonase, PAL. Sometimes they used to ask only this is PAL only. So marker enzyme for ripening process. What is the role of this polygalacturonase? It can degrade the cell wall at the time of a ripening. It is regulated particularly into the cell wall, and for the color changes, aroma production, and the taste and the sensory characteristics of the fruit, which are related with the this enzyme, particularly this enzyme. So all the processes at the time of a ripening, which can be regulated by the polygalacturonase enzyme. Then seed dormancy hormone, it is ABA. This is repeated. Bud dormancy, that is ABA, particularly ABA. These are the options. Stratification is a seed. In a seed, it is a treating seed to stimulate natural condition that seed must experience before germination and can occur to encourage the germination. So it is a pre-treatment before germination. Stress-related hormone ABA, again stress-related protein, heat shock protein. This is repeated. Repeated question. Epigel germination repeated. Rice it is a short day plant. Sugary patch, it is in banana crop. Dieback of apple, potassium deficiency, cell division related hormone. This is a cytokinin. Cytokine, not a cytokinetics, cytokinin. Phytochrome molecular weight, that is a 125 kDa. It is in Tej and Ziegler, page number 494, from where I have answered this question. Then PFR inhibitory effect on flowering of a short day and long day plant, that is in the short day plant. Inhibit the flowering in the short day plant. This is photosynthetic active radiation in the spectrum, that is a 400 to 700. UV protection, it is due to the carotene pigment. Beta carotene, which can remove the oxidative stress. Every stress induces the stress, oxidative stress. Sabhi stress may oxidative stress. You can see that the dot stress, then water, then temperature stress, then freezing stress, heavy metal stress. So in all stress, they are causing the oxidative stress in the plant. So that is why the answer it is a every stress induces the which stress, that is the oxidative stress. Plant which is truly drought resistant. So wheat. Option it is sunflower, fenugreek, and cynodon dactylon. So wheat it is a answer. Light it is important for more for photosynthesis and photoperiodism. No doubt, the light it is very important for photosynthesis and photoperiodism. Circular DNA found in bacteria, chloroplast, and mitochondria. Most usable cytokinin in the media, that is a BAP. Long form of BAP. Sometimes they used to ask the long form. Benzyl aminopyrin. This is a cytokinin source. You can add the kinetin also. You can add the cytokinin other sources also. But result from the BAP, it is more significant in case of a shooting, in case of a callus production. In case of a shooting, particularly in shooting, there is a BAP cytokinin source we use for the tissue culture media. In the MS media, we used to add the BAP. Protoplast means cell without cell wall. Storage temperature for mango, it is a 10 to 12 degrees Celsius. This question, it is 2 or 3 times they have asked in the examination. That in Fahrenheit, sometimes they used to ask in the Fahrenheit, sometimes they used to ask in the degree Celsius. So I have written here in a degree Celsius, that 10 to 12 degrees Celsius. Storage of a fruit and vegetable, it is a cold storage only. Then edible wax used in which fruit quality improvement, that is a carnauba wax, shellac, and resin wax you can use as an edible wax for the post-harvest handling of a different fruit. Then PMA, it is a anti-transparent. So particularly phenyl mercuric acid, it is a fungicide used as a anti-transparent. Gas-related ripening, it is ethylene. Translocation oxime inhibitor, it is a TIBA, iodobenzoic acid, acetic acid. DCMA, it is a inhibitor of photosynthesis 2 system, 240 times more global warming potential of which gas. So you have to search for this question. Where is the life of a cutted gladiolus? It is due to HQS long form at HQS hydroquinoline 200 ppm. It is used for the wage life solution for cutted gladiolus. You have to read the climacteric and non-climacteric fruit and ozone depletion gases, that is a global warming gases, and GA3 and GA1 conversion organelle, which is related to the biosynthetic pathway, because GA, it is not a single element, that is, it is ranges from GA1 to GA59. So you have to go for the GA in detail. Nitrate reductase, nitrogenase, nitrate assimilation. These are very important portion of the nitrogen metabolism. So you have to go in detail for the nitrogen metabolism chapter, and element related to the nitrate reductase, go in depth, so you can answer such type of a question. So
Sorry to interrupt you, ma'am. Actually, we are running out of time, ma'am. So kindly complete the presentation.
there is lot of material, I think so. This is very short material.
Yes, ma'am. Yes, ma'am.
So I think so. Okay.
You may take 5 minutes, 5 more minutes.
Okay. So annual yield or for the biological nitrogen fixation. So it is might be 170 metric ton for the per year. In Tay-Zant-Ziegler, it is a 190, and in Wikigen it is a 170 to 180. Like that they have written. Azula symbiotic with the crop in rice, and the flowering of a pineapple, it is induced by the ethylene and the GA hormone, because both the hormone which are related to the female and male flower proportion. Cyanide resistance pathway, it is regulate the ripening process. Stomatal opening, it is a potassium element, it is responsible for that. For photosystem 2, chlorine and manganese will related. Brassonylate biacetase, it is for the leaf unrolling. Carbonic anhydrase, it is a related with the Kelvin cycle. Element related to the transamination, that is a nitrogen. Drought related protein HSP. Garner and Allard given the photoperiodism. Rhythmic induction given by D. Marin. So there are many questions. So you have to read about the stress physiology, you have to read about the flowering, you have flowering physiology, you have to read about the seed physiology, then post-harvest physiology, and then basic metabolism like photosynthesis, respiration, transpiration, ascent of sap, and uptake, active transport, passive transport, symplastic, apoplastic transport, and Hartmaschner book. Then the books are related with the ARS state and ARS mains, ARS preparation for Wikigen, plant physiology by Wikigen, then plant physiology by Tay-Zant-Ziegler, then Leninger, plant biochemistry by Leninger, then BDC for plant biotechnology, and the DVD and DVD for stress physiology, and these books are sufficient for the preparation for NET preparation. So thanking you for joining me, and thank you organizers for giving me this great chance. This is very important session for the NET aspirant, NET preparing aspirant. So thank you, Vanna.
Thank you so much, ma'am. Thank you so much for your this kind lecture. what I would like to say that your presentation is mind-blowing. The material which you have shown in the PPT will definitely help the students. You have wrote a number of questions that it shows the hard work that you have done for the students. So definitely student will get over your this presentation and will get definitely success in their examination. Thank you so much, ma'am. Thank you. So the last session is about crop physiology. Now the upcoming session is from the genetics and plant breeding. For that, we have Dr. Y. Langthorningambi Devi. She's from IGKV Raipur. So I welcome you, ma'am. Hello.
Yes, hello.
Yes, ma'am. You may join, ma'am. You may join.
Okay. Shall I start?
Yeah, ma'am. You may start, ma'am.
Okay. Is it visible? The screen is visible, right?
Yes, it is visible.
Okay. Good evening, everyone. My name is Lintoy Ngambi, and then I did my PhD from College of Agriculture, ICAR Raipur, and right now I'm working in one project in Institute of Bioresource and Sustainable Development, Manipur. So this ARS NET, it has been conducted by ASRB, which we all know that. So this is an exam which is conducted by ASRB to get this NET certificate, and through this exam also you can like get the job opportunity like those of the SMS, STO. So the recruitment of this SMS, STO, it depends on year-wise. Okay, so like this year, like there is recruitment from for this SMS and STO from the same paper. So preparing for this exam is very useful for getting the NET certificate as well as for job opportunity. Okay, so like it is very much necessary to know this cutoff for different categories in order to pass for this exam. Like different categories have different cutoffs. So this crossing this cutoff means you will be qualified for the NET exam. So cutoff percentage as well as the qualifying mark is given here. Some of the tips and the tricks which you can follow for preparation of this NET exam is, first of all, for preparing any kind of the exam, you must have the syllabus with you. Okay, so first of all, what you can do is downloading the syllabus of the genetics and plant breeding, which is available in the net. So first of all, you download the syllabus and you make you paste your the syllabus just near to your study table. Okay, so first of all is the downloading of the syllabus, and after downloading of the syllabus, you paste it, and then like you make a plan. Okay, plan like for each topic. For each topic, you make us a plan schedule like. From this time I will finish this this much topic. Unit wise is there, so you can first like make a plan schedule for the unit wise, and ultimately you can make this plan schedule for its topic. Okay, so like after making this topic, the next is to search for the materials or like the materials for this for the different topics. Okay, so the basic things to clear all the concept is always to refer the textbook. Okay, so different textbooks which I will be giving in the later slides. So first of all, if you want to clear all the concepts of the genetics and plant breeding, it is very much necessary to to follow and then to read the thorough this one textbook. Okay, so after from the textbook, it is best if you can make your own notes. Okay, own notes, this will be very much easier if you want to go for the revision. Okay, so making your own notes is very much useful. So after making of your own notes, you can for each topic, like before, like you were making a plan schedule, right, for each topic. So for each topic, you can, like, after finishing of one topic, you can start solving questions, MCQ questions for that particular topic. Okay. So after this one, like, try to solve as much MCQ questions for related to that particular topic. Okay. So while solving the questions, what I want to say is that, like, what we do is that we always, like, if we know the answer, we always choose the right answer and leave behind this one other options. Okay. So, like, if the question is new to us and if we know the like correct answer, but like other options are there. Okay. So here we can use one more thing, like the other options, the terms, if it is given for the other options, you can also study those, like other option terms. Okay. Like next time the other options one, it may come, right? So, like, take this as an advantage, like don't just leave it at just. So you can study the this one, the related to the other topics also. Okay, other options also. So what you can do is try to solve as much questions as possible. And then, like, for us, we have different newly released different varieties, different genes are present. So for different crops, right? So you can actually make a chart, and then, like, for crop wise, you can make a chart in order to make it, like, more easier for you to remember. And also it will be very helpful for quick revision. Okay. So you can make crop wise, like crop, it can be self-pollinated, cross-pollinated, and then often cross-pollinated, as well as the their ploidy level. You can, like, include whether they are diploid, tetraploid, hexaploid, and all. So in a one neat table, you can make for its crop wise, their varieties and all. So these important questions, it used to come in exams, so it will be very helpful. And then, like, always try to give trial exams in the sense, like, if you want to, like, if you're in the MSc first year, then you can start giving this NET exam from now itself, because next time after finishing MSc, it will be considered that we know, but like as a trial exam, so that you can assess your performance, as well as, like, you can improve, and then you can know what kind of the question pattern is coming. Okay, so these trial exams you can give. And then one most important thing is that do not forget to revise whatever you have studied. So if you forget to this one, go for revision of whatever you have studied, then, like, half of the things you used to forget. Okay, so don't forget to go for the revision. So these are the tips and the tricks which you can actually follow for a beginners. Like, if you want to prepare for the NET exam from now itself, and if you have time, then you can, like, you can actually follow this slow method, and it will be very effective. Okay, but if you have, like, very less amount of the time, and you want to extract that much little amount of the time in order to get a very big impact, then you can, like, you can start studying for these important topics. Okay, like, these are some of the important topics from which, like, most of the questions, measure of the questions used to come. Okay, these topics you cannot leave behind, because these topics are very much important for genetics and plant breeding. So there is Mendelian inheritance is there, like law of Mendelian inheritance is there, that we all know, and its extension is, like, the different types of the deviation. Okay, deviation from the Mendel's ratio, which we also call as the epistasis. Okay, so different types of the epistasis is there, like complementary, supplementary, and then this duplication and all different types are there. And next is the cell division, like different cell division type, like mitosis is there. Meiosis is there. So in mitosis also different stages are there. So each stage, like, you have to know its stages and what happens in that particular stage. Okay, so these points you have to know. And then linkage is there, and then, like, different types of coupling, and linkage is there, and then, like, calculation of the linkage map is there, and calculation of the distance between two genes. Those things are important. And the DNA replication, transcription, translation. Okay, so in this, like, different steps present in replication, transcription, translation, as well as the enzymes, okay, enzymes, and then which is like responsible, which are present in different stages, you have to know. And then gene code is there, that also you have to know. And for the mutations, like you have to know like different types of the mutations. There is physical, chemical, and then in the physical also, like different is ionizing radiation and all different types are there, that with example, okay, with example you have to know, right? And for apomixis is there, apomixis also different, the types of the apomixis is there that you have to know. And self-incompatibility, different types of the self-incompatibility is there, sporophytic, gametophytic, and also like, and then the crops or the families which belong to that sporophytic and gametophytic, that you have to know, okay? And then male sterility, different types is there, and then different types of the crosses, like single, double, top crosses, polycrosses, and as well as their formulas, okay, how to get that kind of the crosses and all, like these formulas you have to know. And some of the important scientists and their discoveries along with the year that you have to know. And class of the seeds, like those we have this nuclear seed, breeder seed, foundation seed, certified seed. So those seeds you have to know, and along with the tag color. And then chromosomal aberrations is there. And then ploidy level, like aneuploid, polyploid and all, like different types of this ploidy level is there, that also you have to know. And different mating system and design you have to know, and Hardy-Weinberg law, and how to find out this gene and the genotype frequency, as well as QTL mapping is there, and the sequencing method. And also you have to know the breeding methods, okay, breeding methods which we followed for this for resistant breeding, for abiotic stress, and the nutritional quality, okay. And then there is different types of the mapping population, like F2 mapping population, reals is there, and then like nil is there, and then like there's and different types of the molecular marker is there, like AFLP, and then this RAPD, SSR, and then SNP, ISSR, and all. And then like you have to know, okay, its marker, what is their characteristics and all. Excuse me. And then you can know for different hormones, like different hormones are there, no, auxin, gibberellin, cytokinin, and all. And then so each hormone you have to know, as well as their functions, okay. And then like different breeding methods are there, which we followed for like self as well as the cross-pollinated crop. So different breeding method which we use for self, cross-pollinated, as well as for the improvement of the population breeding. So different breeding methods are there, okay. So that you have to know, like this breeding method, when we have to use and in which crop you have to use, okay. And then like different statistical design for conducting of our experiment, okay. So for us, like for conducting of our experiment, the germplasm we have, it can be grown in different types of the design or experiment, okay. So those statistical methods like CRD, RVD, and then this augmented lattice, those things you have to know. And then this brassica U triangle is there. So every year it used to come this one. So these topics you can mainly focus if you have the limited amount of the time. And these are some of the books which is very common, and I think which is known to everyone, but it is very important and must to have books, okay. So apart from these books, there are some other books which is really necessary if we want to clear our concepts, okay. So for quantitative genetics, you can follow this biomedical methods in quantitative genetic analysis and the biomedical techniques in plant breeding. And for genetics, this Nostad and Simmons one is very, very basic, and then the way they have written is very basic and it's easy to understand, okay. These books you can follow when you want to clear your concepts, okay, because after this NET, if you're preparing for ARS also, then like clearing the concept will be very much important, because in ARS mains you have to write a subjective type, not objective type. So there, like the way of your expression will know how much you understand the topic or how much basic you have known regarding the topic, okay. So this genetics, Nustad and Simon, and then there's Gardner, principle of genetics. And then for nowadays, the questions, many of the questions used to come from this molecular reading, like those markers and this mapping population and all. So for that, you can actually refer this marker assisted plant breeding principle and practices. This is very good book. So here, like you can refer those molecular part, and then molecular plant breeding UNBG also, it's very good. Okay, and then like for biotechnology part is there. So for that, you can refer one biotechnology book. So that's all I would like to say. And then like lastly, I would like to thank this Just Agriculture Education Group for organizing such an informative and useful session for the young aspirants, which I think will be very much useful for them. And to all the aspirants and students, I would like to say, prepare nicely and all the best for your future. Thank you.
Thank you so much, ma'am, for your this time and your lecture. Ma'am, just like we now this time we have a very short of time for the examination. We have just only, I think, 10 to 12 days.
Yeah.
So what tips you will give to the candidates? Like the syllabus is very huge. So
yeah, that only I was saying, like if you have limited time, like I would like to refer those topics which I have mentioned, plus this objective type. Okay, so objective types, like if you go through the objective types, then also like this one, like most of the questions they used to come this from this objective type. Okay, so like you can refer both objective type and some and the topics which I have mentioned before. Those are very important and basic one. So those like referring those topics, like it won't take much time, because I was because those topics were like only a small part of the syllabus. Okay, so those topics they can focus on those topics, as well as plus multiple choice. Multiple choice is very much important to clear the NET exam, not for ARS. Okay, like for ARS also it's important, but for the NET exam, the objective types, objective question bank, MCQ question bank, it's very much necessary. So like along with that, this one they can follow.
thank you, ma'am. And ma'am, any suggestion for the attempting the examination? Any suggestion for attempting the examination while the candidate is sitting in the examination? So what will you suggest for the?
Like what I want to suggest is that, like while giving the exam, this one, like if after like while giving the exam in the first attempt, I want them to do only the questions which they think is 100% sure. Like after doing those questions, if they can count the questions and they will know how much they did. Okay, if it crossed the cutoff, which I have mentioned before for different categories, if they know they have crossed the cutoff, then like they don't have to risk it, because sometimes what we think is that we used to be very like greedy and we used to do for more kinds of the questions which we think will be correct, but at last like it used to be like negative impact for us, like many negative marking used to go. So what I want to say is that first of all, you do only the 100% or only the questions which you are 100% sure, and those after that, if you think you do not cross the cutoff, then you can go for this blind hit, not blind hit, like which you think 50% like correct. Okay, so those things you can attempt, which is something which I would like to suggest to the aspirants or students who are like appearing for this NET exam.
Okay, thank you so much, ma'am, for your valuable time. Thank you, ma'am.
Thank you. So can I leave?
Yes, ma'am.
Okay, thank you so much.
So at last, I would like to thanks all the respected speakers for day 4 session of ICAR NET series. Just one suggestion from my side, that while you go for attempting the examination, if you think that you have cleared the cutoff, like in general category, you have attempted the 60% questions. So don't go for more than 20% guesses out of total. Okay. Yeah, you may clear it on 50%. But when in examination, if you have time, then sometimes what candidate do, they attempt all of the questions, like they have time. So be patient and be calm and be all the best for your examination. And thank you so much.